Hey guys, welcome back. For those who are new to the channel, my name is Teddy, and this channel we will break down exam questions and discuss related concepts to prepare you for exam. And if you like the content of my video, please subscribe to the channel so that you will get a notification as soon as I release the next part. And in this part we will talk about antibody molecule and diverse generation in T cell and B cell receptors, specifically for antibody. Let's get started.
First question. An immunologist is examining the role of antibody regions in immune system interactions. The Fc region of an immunoglobulin molecule is crucial for which of the following functions? So he's studying antibody and its interaction in the immune system. So the Fc portion, what's the role of the Fc portion? Recognition and binding of antigens. Recognition and antigen binding is by FAB, so this is not the right. Interaction with Fc receptors on immune cells. Yes, the Fc portion interacts with Fc receptors. That seems to be the right answer. Neutralization of pathogen. No, neutralization is after binding the antigen, which means through FAB. It's not right. Determination of antigenic specificity or determination of antigen specificity. So the antigen specificity is determined by the part of antibody molecule which interacts with the antigen. So this is not right. So I think the correct answer will be B. So let's see. B. So antibody have different structural components, so in two dimension, and to make it simple for understanding, it can have a Y-like structure like this. So this is FAB, or it have a function of antigen binding, and this is FC. It have function of crystallization. So the FAB portion, or these two portions, will bind or will interact with the antigen, so they have function of antigen binding, or in short they are called FAB. And this long part is function of crystallization after it's treated with an enzyme. So generally antibody have different components. This large structure is a heavy chain and the small structure is a light chain. So an antibody molecule have a heavy chain and a light chain, and this heavy chain and light chain are connected with each other by a disulfide bond, and there are also globular regions within the heavy chain and light chain which are interacted or connected with a disulfide bond. In both light chain and heavy chain have variable region and constant region. The variable region is on the FAB part and the constant region is on the FC part, but the FC part is made up of the heavy chain. The light chain is not going to be part of the FC region, but it still have its own constant region. And both heavy chain and light chain have globular regions in 3-dimension, and those globular regions can be visible by 3-dimensional imaging. But in 2-dimension, to make it simple, it's like a Y-like structure or Y letter. So these two FAB regions will combine or connect with each other and form a hinge region or a neck region. So the connection site or the interaction site of those two FAB regions is called a hinge region. Some antibody molecules have large or long hinge regions, some have short hinge region. And in addition to that, on the FC portion, on the second constant heavy chain region, there is a sugar molecule which will increase solubility of the antibody molecule, and we call that oligosaccharide. So these antibody molecules can be divided into isotype, allotype, and allotypes. So the isotype is type of antibody molecule. So based on the major amino acid change on the FC region, we can group them into five types of antibody molecules: immunoglobulin M, immunoglobulin D, immunoglobulin G, immunoglobulin A, and immunoglobulin E. So there are 5 types, but in some immunoglobulins there are subtypes and we call them allotypes. This is a minor amino acid change on the heavy chain region, on the FC region again. So these allotypes are for example, for immunoglobulin G or immunoglobulin G, it have 4 allotypes and from 1 to 4. immunoglobulin G1, G2, G3, G4. And for immunoglobulin A, there are two types, alpha 1 and alpha 2. And for light chain, we have two major types of light chain, kappa light chain and lambda light chain. Around 60 to 65% of the light chain is a kappa in our body, but lambda light chain have allotypes 1 up to 4. Lambda 1, 2, 3, 4. And there are also idiotypes. Idiotype is majorly on the FAB portion of the antibody, and since it interacts with a specific type of antigen, is diverse in a type. So these diverse types can also be typed into idiotypes. So antibody molecule can be treated by an enzyme and will give different products. If it is treated with peptide, it's going to break the antibody molecule before the hinge region, and it will give two separate FAB regions and one FC region. So it will be broken down into 3 fragments. So that cannot agglutinate an antigen because a single binding site is separated by itself, so it cannot bring 2 antigens in a close proximity. But if it is treated with pepsin, it will break it after the hinge region, and the FC part will be degraded, and it will be left with connected FAB regions. It's not going to have immunological function, but it can agglutinate antigens because these 2 antigens can be brought together or in a close proximity by 2 antigen binding sites. The antibody molecule have 4 major functions. The first one is neutralization. Neutralization is effective immune defense mechanism for toxins and small antigens like viruses. And the other is complement fixation. It can also activate the complement system. And the other is opsonization. Opsonization means facilitating the process of phagocytosis. So antibody molecule is good in opsonization. And the other is ADCC, or antibody-dependent cell cytotoxicity, especially for NK cells. Once an antigen is bound or a cell is bound with an antibody molecule, since a natural killer cell have Fc receptor, they can interact with the antigen easily and will facilitate cell cytotoxicity using NK cells. So we call that ADCC. So let's move to the second question. Which immunoglobulin G subclass is most efficient at crossing the placenta to provide passive immunity to the fetus? So the only immunoglobulin which can cross the placenta to provide protection for the fetus is immunoglobulin G. The rest will not cross the placenta. Why only immunoglobulin G? Because it have a binding site. We have immunoglobulin G1, G2, G3, G4. Which one is best in crossing the placenta? That's the question. So G1 is the best to cross the placenta, but the other are not that much effective in crossing the placenta. Let's see the answer. So different allotypes have different features. For example, when we talk about crossing the placenta, G1 is more effective in crossing the placenta. But when we talk about complement fixation, it's a different story. So G3 is best in fixing the complement system. Next to that we have G4. Next to that we have G1, then G2, G4. G4 is weak in complement fixation. And when it comes to crossing the placenta, G1 is most efficient, then G4, then G3, then G2. So different immunoglobulins have different functions or different effectiveness in different set of immune response. For passive immunity, G1 will be the best one. And again, when we talk about other types of immunoglobulins, again, when we talk about immune protection through complement fixation, immunoglobulin M is better for opsonization and complement fixation because it's large in size, forms a pentamer structure or 5 immunoglobulin M molecule in a soluble form, or in a secretory immunoglobulin, 5 immunoglobulin M molecules are bound together by a joining chain at the center. So that large size gives it a better capability of agglutinating an antigen, complement fixation, and opsonization. And Next to that, we have immunoglobulin E, which have receptor on eosinophils and mast cells, which enables this type of immunoglobulin to activate inflammatory reaction and allergy or mediate allergy. And immunoglobulin A, it can be formed as a dimer in a secretion, in a secretory form, not membrane bound. So it can be a dimer, and it's resistant for enzymes in the mucous membrane, and it can be secreted into a secretory parts or mucous membrane due to its secretory piece or bind interaction with the secretory piece. It also J-chain since it forms a dimer. Immunoglobulin D, its function in the secretion form is not well known or elucidated, but it is one of the first immunoglobulins which appears on the surface of a B cell during maturation. During B cell maturation, first you will express immunoglobulin M, then immunoglobulin D. Then after completing a maturation and later activation, it may have class switching to other types of immunoglobulins. So this is just the highlight of immunoglobulins. Let's move to the third question. So your patient became ill 10 days ago with a viral disease. So 10 days ago, there is a viral infection with our patient. So laboratory examination reveals that the patient's antibody against this virus have a high ratio of immunoglobulin M to immunoglobulin G. What's your conclusion? So there is high concentration of immunoglobulin M than immunoglobulin G. Basically, we will expect large concentration of immunoglobulin G. It is the largest. In concentration, immunoglobulin G is the largest. Okay, next to that immunoglobulin A, next to that immunoglobulin M, then immunoglobulin E. D is very minimal. It's not that much present in secretion. So the first option is it is unlikely that the patient have encountered this organism previously. Okay, it's unlikely that the patient have encountered this organism previously. Means is encountering the pathogen for the first time or primary response. This seems to be right. The patient is predisposed to immunoglobulin mediated hypersensitivity reaction. It has nothing to do with IgM and IgG. The information given is relevant to previous antigen exposure. No, it is relevant. And it is likely that the patient has an autoimmune disease. No, this is not linked with that. So A is going to be the correct answer. Okay, A.
So there is a variation in concentration of immunoglobulin molecules normally in the plasma, and they also have different half-life in the blood circulation. So the highest concentration of immunoglobulin is immunoglobulin G normally, followed by immunoglobulin A, immunoglobulin M, and immunoglobulin E. So immunoglobulin E is the lowest in concentration, and immunoglobulin G is the highest. And also the half-life is in similar order. Immunoglobulin G have the longest half-life, followed by alpha and mu, and the immunoglobulin E have the shortest half-life, which is around 2 days, and for immunoglobulin G it's around 20 to 23 days. And this long half-life is due to a special receptor on the membrane of endothelial cells, so neonatal Fc receptor, which will bind with immunoglobulin G, and immunoglobulin G will be taken up by endosomes into the endothelial cells. Then it will stay there and will be protected from degradation due to this special receptor or neonatal Fc receptor. Then it will be recirculated in the circulation, which prolongs its shelf life or half-life. So in this question, it's asking what is the message when there is a variation or when there is higher concentration of immunoglobulin M than immunoglobulin G, which is not normally happening. So due to presence of immunoglobulin M and immunoglobulin D on the surface of a B cell, which is a naive B cell, as a B cell receptor, when a B cell is activated for the first time, it's going to produce immunoglobulin M. So primary infection or primary immune response will have higher concentration of immunoglobulin M than immunoglobulin G. But during secondary response, or when we are exposed to the same antigen for the second time, since we have a memory from the previous exposure, we will have higher concentration of immunoglobulin G than immunoglobulin M. So in our case, since there is high concentration of immunoglobulin M than immunoglobulin G, it can tell us that the person is infected for the first time and it is an acute infection. So when there is high concentration of immunoglobulin M, it indicates acute infection. When there is high concentration of immunoglobulin G, it could be a previous infection or an immunization or a chronic infection. So different concentration of immunoglobulin molecules have different meanings. So let's move to the next question.
Okay, fourth question. During the process of VDJ recombination, which of the following statement about the heavy chain gene is true? So it is during VDJ recombination or during diversity generation and heavy chain gene. Okay, the gene segment V, D, and J are randomly rearranged to create a unique antigen binding site. So they are randomly selected to create a unique antigen binding site for the variable region. This seems to be true. The antigen selects the specific V and J segments for recombination. No, the antigen will not select. Well, this VDJ recombination is before antigenic exposure. It's during maturation of these cells. So this is wrong. The antigen will not do that. But for B cell, when there is somatic hypermutation in secondary lymphoid organ, it's not going to select that, but the B cell will try to fit its receptor with the antigen. Only one V region segment is available for recombination with multiple J segments. No, there are multiple V, multiple D, multiple J segments, and this is again wrong. And the heavy chain gene does not require D segment to form a variable region. Again, this is wrong. But the light chain does not require a D segment. So VDJ is for a heavy chain. So seems the right answer will be A. Okay, so in here, the adaptive immunity is diverse, or diversity is one feature of the adaptive immunity, and there is a process of diversity generation in B cell and T cell receptor development. So it have multiple factors will contribute for this diversity generation. The first one is
the receptors are coded by multiple germline genes.
And for the variable region, for the heavy chain, as well as for the beta chain of T cell receptor, it's coded by 3 segments: the variable segment, diverse segment, and junctional segment, or VDJ. For the light chain, there is no D segment or diverse segment, and it's coded by V and J. So for this difference, there are multiple genes. For example, in human, the heavy chain variable gene have 45 locus, and the D have 23, the J have 6, or we have 23 diversity segments, we have 6 junctional segments, and we have 45 variable segments. For the kappa, we have 35 variable segments, we have 5 junctional segments. For the lambda, we have 30 variable segments and 4 junctional segments. And again, the heavy chain, kappa chain, and lambda chain are coded in different chromosomes. For example, the heavy chain is coded in the 14th chromosome, 1-4, and the kappa is coded on the 2nd chromosome, and the lambda is coded in the 22nd chromosome. So multiple genes will combine to create one receptor. So this is one factor. The other factor is combination of these variable, diversity, and junctional segments of a gene will create its own diversity, because they are not going to be precisely cut in determined location. So there is also diversity by combining this variable segment, diverse segment, and junctional segment. So for the heavy chain, one randomly selected V segment, one randomly selected D segment, and one randomly selected J segment will be recombined first, then they will recombine with the constant region. So the constant region is not that much variable, it's not diverse. For example, for the heavy chain, we have 9 constant regions. For the light chain, we have 5. For the light chain, the VDJ segment will recombine first, then it will recombine with constant chain. So this combinatorial joining of the VDJ and VJ segment will create its own variation. So how does it cut it? Because there are signaling sequences, recombination signal sequences on the DNA, and the enzymes, specifically RAG1 and RAG2, or recombination activating gene 1 and recombination activating gene 2 enzymes, will recognize these RSS areas, then they will cut the DNA. So they are endonucleases. So when this endonucleases cut the DNA, they will create a cut region, and next to that, this repair enzymes or double-stranded DNA repair enzymes will try to fill the gap by adding palindromic sequences or nucleotides, and we call that P addition. But since it may not be able to fill the whole gap by adding palindromic sequences, it may also introduce additional nucleotides from 5 up to 15 nucleotides additional. So we call that N-nucleotide addition. So this is additional diversity again, random nucleotide addition of N-nucleotide addition as well as palindromic addition. And the other is junctional diversity. Again, previously I said that when it tries to cut the sequence or the DNA, it's not going to be precisely the same area for different cells or for different tries of creating the VDJ recombination. So it will have variation, then we call that junctional flexibility. Again, after it forms the heavy chain and the light chain, it will recombine the heavy chain and light chain, and it also creates additional diversity. So these all factors will contribute for diversity generation in T cell and B cell receptor. But for B cell receptor, once it have this B cell receptor within the primary lymphoid organ, it will migrate to secondary lymphoid organ. Then it will wait for the antigen. Once it interacts with the antigen, since it may not have a perfect fit for the antigen, it will try to edit its B cell receptor with high rate of mutation. Well, technically speaking, it's going to do the same type of recombination or VDJ recombination and VJ recombination that happened in the primary lymphoid organ, but it is after antigenic exposure. So we call that somatic hypermutation. And it happens within the secondary lymph. During B cell development, Allelic exclusion ensures that a B cell expresses only one type of immunoglobulin heavy chain. Which of the following accurately describes the mechanism of allelic exclusion in heavy chain gene rearrangement? Okay. Both alleles of heavy chain gene are rearranged simultaneously, but only one allele is expressed. No, they are not simultaneously expressed. It will try one after the other. The heavy chain gene rearranged on one chromosome, then the other allele is permanently silenced after successful rearrangement. This is correct. So it will try expression of the gene on one chromosome, and if it fails, it will try the other. But if this one is successful, it will suppress the other permanently, because the B cell is going to have one and only one type of antibody based on its idiotype. Okay, it's not by allotype and isotype. Based on idiotype or antigenic binding site, it's going to have only one type. So once the first one is successful, it's going to dump the others. But if it's not successful, it will try the others. So this it seems to be correct. Both alleles of heavy chain gene are actively rearranged, but only one is retained for expression. No, this is wrong. It may have multiple constant regions, but it's not going to have variable regions which targets multiple antigens. The light chain gene rearranges first, leading to the expression of only one heavy chain allele. No. So the heavy chain is expressed first, then it will be expressed on the surface. The heavy chain is rearranged first, then it will be expressed on the surface of a B cell with surrogate protein, then pre-B cell receptor. That's the name of a pre-B cell receptor, or that's the name of the receptor. Then it will send a signal. If it's a functional heavy chain, then it will move to the light chain. Then it will form the light chain. For a T cell, the beta chain will be formed first and will be expressed on the surface of a cell. Then it will be tested. we call that free T cell receptor. Then we will move to light chain or the alpha chain expression. So that is allelic exclusion. So allelic exclusion makes sure that T cell and B cell recognize only one antigen, because the other variable chain rearrangements will be suppressed once it have effective receptor produced. So that's about allelic exclusion. And hopefully this session was helpful for you during your exam preparation or to refresh your knowledge on basic immunology. And until we meet next time, see you.